will make a start in 1 minute. So please be patient. Thanks for joining us this morning. Okay, I'll begin. So you're very welcome. Good morning, good afternoon, and good evening to everyone, wherever you are in the world today. My name is Sinead Leahy. I'm the Principal Scientist at the Ag Emission Centre based here in Palmerston North in a very wet Manawatu of New Zealand. I'd like to welcome you to our State of the Science webinar series. This series is run in partnership with the Irish Teagasc Climate Centre, and it's a fantastic opportunity to bring researchers from both countries together to sort of share knowledge, insights, and some of the latest developments. We're delighted that we actually have so many in our audience today. It's a big webinar today, so thank you for joining us. And also, please note during the rest of the year, we do have a number of other exciting webinars planned, so please keep an eye on our socials for more information. Today's webinar is all about microbes and methane. And as a semi-retired rumen microbiologist, I'm very excited about this particular subject. And we're actually focusing in on lactic acid bacteria, which is even more exciting because these are one of my favorite groups of microbes that exist. I'm going to be your chair today. And before we get into it, I just want to give you some brief housekeeping. I'm going to let both our presenters complete their presentations before we move into a Q&A session. Questions can be submitted using the Q&A button. You should see this at the bottom of your screen. Please feel free to submit your presentations or your questions at any time during the presentation. If you see a question you're interested in, you can give it a thumbs up. That helps me prioritize the questions that matter most to the audience on the webinar today. A recording of today's webinar will typically be available via the Ag Emissions YouTube channel within 1 week. So if you miss anything, you'll have an opportunity to go back and hear our presenters again. So let's kick off. And first up, I'd like to welcome Dr. Graham Atwood. Graham is a principal scientist at the Newly Bioeconomy Science Institute and a leading expert in rumen microbiology. His work has been instrumental in advancing our understanding of the microbes responsible for methane production in ruminant systems. And we're delighted to have Graham here today. On a personal note, Graham was my first boss in New Zealand, and he's one of the reasons, sadly or greatly, that I'm still here in New Zealand. So it's really, really lovely to have be chairing Graham's presentation today. He's also a very keen fisherman and on occasion will catch some fish. So Graham, welcome. Over to you. Please share your presentation and we'll kick off.
Thanks, Sinead. How's that looking? Can you see that?
Yeah, that looks great, Graham. Over to you.
Okay, thanks very much, Sinead. And thanks to the Ag Emissions Centre for the invitation to speak with you about lactic acid bacteria and their potential usefulness in mitigating ruminant methane. Like Sinead, I'm a fan of lactic acid bacteria. They're incredible microbes and they have a whole range of potential uses. So ruminants and methane emissions. So ruminants are really important to in sustainable agricultural systems, but they also are important sources of greenhouse gas emissions. Methane is the largest source of greenhouse gas emissions from ruminant production, with over 90% of emissions originating from indirect fermentation. That's from the digestive processes that take place in the rumen there. The livestock farmers are facing challenges of being able to carry out efficient, sustainable food production while moderating or reducing greenhouse gas emissions. And new technical mitigation options to reduce methane emissions are urgently required. And the question here, and what we've been working on for the last few years, is determining or looking at the ability of lactic acid bacteria to be used in methane mitigation strategies. So what exactly are lactic acid bacteria? Well, they're gram-positive, acid-tolerant, facultatively anaerobic bacteria that produce lactic acid as a major end product of their fermentation of carbohydrates. They're widely distributed in the environment, and they mediate fermentations and dairy, meat, and plant products. Biochemically, they include homofermenters, which mean they produce mainly lactic acid as an end product, or heterofermenters, which also give other end products such as acetate, ethanol, and carbon dioxide. They have a long history of use in human and animal foods, which has resulted in most LAB having qualified presumption of safety status in the EU, or generally recognised as safe status in the US. And the main LAB genera that are used as starter cultures are Lactobacillus, Lactococcus, Leuconostoc, Pediococcus, and some species of Enterococcus and Streptococcus. Now, some really enthusiastic bacterial taxonomists have recently divided the lactobacilli into 25 different genera now. And if people want to find out the details about that, I encourage them to dig a bit deeper into the bacterial taxonomy. Lactic acid bacteria are actually already used on farms for a number of uses, and as direct fed microbials, as probiotics, and as silage inoculants. And their claimed benefits are wide and varied. And what I've done is listed a few there. They're particularly useful in neonatal calves. They reduce the incidence of diarrhea and fecal shedding. And as the animal develops, they are involved in promoting ruminal development. They've been linked with increased feed efficiency and body weight gain, and a variety of other uses there. But in general, their main use has been in neonatal ruminants, and mainly targeted towards health benefits. But lactic acid bacteria also live in the rumen, so they're found there naturally as a part of the normal ruminant gastrointestinal tract microbiota. And they're especially prominent in young animals before the rumen is fully developed. Although LAB can't break down the structural carbohydrates in plant material, they're very good at fermenting soluble sugars that are released by other organisms during the digestive process. Lactobacillus ruminus and Strep equinus are true rumen inhabitants, so they are known to live there all the time, and Strep equinus is reasonably prevalent and abundant. And it's quite easy to isolate different types of lactic acid bacteria from the rumen. Now, there's a slightly different flavour of lactic acid bacteria in the rumen, and two of the representatives are Candelaria vitulina, which used to be called Lactobacillus vitulinus, and Sharpea azabuensis. And now both of these organisms, although they're more distantly related to the lactobacilli, they perform the same function of producing lactic acid in the rumen. And these organisms are relevant to our discussion on methane emissions because both of those organisms are linked or significant components of the rumen microbiome in animals which have been bred for low methane emissions. And the diagram there shows you how we think these organisms are involved in that low methane emission status. So the Sharpea involved, we believe, are involved in the rapid fermentation of hexoses to lactic acid. And then another organism called Megasphaera ferments that lactate through to butyrate. Now, that particular metabolism produces slightly less hydrogen than the conventional metabolism shown on the right there. And less hydrogen leads to less methane. And we think that's behind the roughly 20 to 25% decreased methane emissions from these low methane yield animals. Now, the effects that LAB have on ruminant methane production is not new. There's a whole raft of studies in vitro which have indicated that lactic acid bacterial strains themselves or the bacteriocins that they produce can have effects on methane production in vitro. And this here just lists all those various studies which have been reported. There are a smaller number of in vivo trials. Lactobacillus pentosus D31 has been shown to decrease methane in sheep, and niacin has also been shown to have the same effect. And then another plantarum strain has been shown to reduce methane. Now, one of the problems with these in vivo studies is the smaller number, and the number of animals that have been used in these studies has been small. So the data isn't particularly convincing when it comes to the in vivo evidence for reduction of methane. And the other approach has been taken to using LABs is to use them to ensile plant material, because they're also involved in that process naturally. And in one study by Cow et al. In 2010, they showed that ensiling plant material and then feeding it to animals reduced methane emissions, in sheep in this case. So how are these lactic acid bacteria having their effects in the rumen? So there's three possible routes that they might be acting. So either by the stimulation of lactate using microbes to produce propionate, leading to less acetate and then less hydrogen, which in turn leads to lower methane emissions. Or directly by inhibition of methanogens, that's a possibility. Or thirdly, by the modulation of other microbes which are involved in producing the substrates which methanogens use. So the formation of hydrogen or formate, or the methoxylated compounds which the particular types of methanogens use. Now, previously there was little evidence that the LABs or bacteriocins themselves could affect methanogens directly. But recent work in our lab has shown or developed a direct assay, methanogen growth assay, which allows us to screen extracts and cultures in high throughput assays. And that's allowed us to screen a much larger variety of strains and extracts for when we're looking for anti-methanogen activities. The availability of this assay has then led us into a collaboration with Catherine, the other speaker on this talk, and Paul Ross at UCC in Cork, and Chagas in Cork as well, but also with other collaborators at INRA and Wageningen and Sacco in Italy. So that program was looking at refining direct-fed microbes and silage inoculants to reduce methane. And we took a approach of screening strains for methane reduction, incorporating those into product prototypes, and then evaluating their efficacy in animal trials. So our screen, our high throughput microtubulate-based screen was essential in screening the strains against methanogens. In this case, we developed the screening assay against Methanobrevibacter bovis coriniani. Now, this is a rumen bacterium, a rumen methanogen which produces methane. It maintains a small population in the rumen, but its main feature is its ability to use ethanol instead of hydrogen and CO2, as well as hydrogen and CO2 to make methane. Now, this particular biochemical quirk or microbiological quirk allows us to grow this organism much more easily in microtubulates and under anaerobic conditions. And once we can do that, then of course, we can screen 96 cultures at a time for evidence of methane inhibition. And so using this assay, we then set about screening over 200 different supernatants and strains from the collective strain collection between Chagas and UCC and AgResearch. And what the screening showed, there was 39 strains which had greater than 50% inhibition. There were 12 strains which showed greater than 80% inhibition of the JH1 strain, or methanogen strain. Interestingly, there were a number of strains which showed not inhibition of methane production, but a stimulation of the growth of JH1. Now, these are really interesting because well, we don't understand how they stimulate methanogen growth, but they could be potentially useful in our ongoing attempts to cultivate more and novel methanogens. And then the other thing that we noticed was that some of the strains, LAB strains, actually produced more than 100% inhibition, which suggested to us that these extracts or these strains were able to actually lyse the inoculum that we were putting into the assay. So that was a significant finding as well. So from those screenings, we were able to come up with a short list of Chagas and UCC strains or AgResearch strains, or in fact a SACO strain, which were, if you like, top of the leaderboard in terms of the inhibition of methane methanogen growth. So from these, they're from a variety of different sources, and they had a variety of different predicted biosynthetic clusters based on their genome sequences that were available. But from the ones that we screened and that we found activity, we were very interested in the LP58 strain from our SACO partner in Italy, which showed almost complete knockout of JH1 growth. So because this strain is commercially available in large amounts, this was very convenient for us to use for further testing in sheep. So that's what we chose, and we moved on to do that. So to cut this a bit short because I'm running out of time, we screened the LAP culture in supernatant and made silages from that strain, or mini silages, and then screened them in vitro. What we saw was the LP58 culture and supernatant increased propionic acid and decreased the acetate to propionate ratio, indicating that it had a particular effect on rumen fermentation in vitro. There wasn't much effects on gas production or methane production. When we then looked at the microbiome of those in vitros, we showed that the bacterial community was slightly enhanced in terms of the Fibrobacter. These are fiber-degrading organisms, and that another group of organisms, the Corynebacteriaceae and the Clostriales, were decreased. And then in the archaeal community, we saw that the culture on the supernatant actually increased Methanobrevibacter abundance while decreasing Methanomycelicoccales, that's a methyllotroph. And then the mini silage decreased Methanosphaera abundance and the Methanomycelicoccaceae abundance as well. So we scaled up the silage making with the LP58. Here's various images of that happening out at our Aorangi farm. And we had a crossover design animal trial feeding these silages to the sheep for 24 days, and then collecting samples and analysing methane, and then collecting samples, crossing over, and then repeating it with swapping the treatments to the different animals. And we had 12 sheep per treatment group. So in a nutshell, this is the methane emissions that were analysed from that trial. Now, overall, there was no impact of the LP58 silage on methane production from the sheep compared to the control silage. And the methane emissions were in the kind of range we'd expect to see from sheep fed a silage diet. But however, there were effects of both the silage and the LP58 silage relative to controls, and in terms of increased propionate production and butyric acid production. And again, we saw that acetate to propionate ratio being affected as well. But the really interesting thing for us was, although the bacterial and the archaeal communities in general didn't change, we saw specific effects on Methanosphaera. These are the methylotrophic methanogens that we had some evidence of inhibition in vitro. We saw this specific effect happening in the animals as well. So although the animal trial didn't show reductions in methane, this indicated that we were having specific effects on a subset of the methanogens in the rumen of those animals. Now, briefly, if I have time, Sinead, I'll just summarise what we've been doing more recently in the rumining program, which is looking at lactate metabolism in high and low methane yield sheep. Now, this background is basically saying, summarising what we know about those low and high methane sheep, so the microbiome in those animals, and basically cutting a long story short, we think that there's a rapid fermentation of those animals mediated by a couple of different ruminant types. One of them is dominated by Sharpea, and that leads to, as I said before, lactate formation, which then goes through to butyrate formation, which leads to less methane. Now, we're really interested in understanding in more detail that both the metabolism involved and the organisms that are driving that, not only the Sharpea, but what other types of organisms are involved in that process. So we have one PhD student, Josh Joseph, which has been looking at directed cultivation of ruminant microbes from the low and high methane animals. And so far, he's been able to isolate 99 pure cultures from those animals. And he's looking at characterizing them. And from their lactate production and utilization, it's hard to determine at the moment what the involvement of lactate dehydrogenases is. So what he's been doing is looking at the genome sequences of cultivated organisms so far, and looking at the pattern of the genes that they have, which encode various aspects of lactate utilization, to see if you can use those gene abundances or distributions to figure out whether they are users of lactate, producers of lactate, or are both users and producers of lactate. Now, the other PhD student we have working on this project is looking at the measurement of lactate pools and fluxes in the rumen in the low and high methane yield sheep. This work has been done by Samuel Muturi. And basically, when we looked at that, we looked at VFA production in those animals, and we showed that butyrate was elevated in the low methane sheep. We also noticed that branch chain fatty acids were decreased in the low methane animal sheep, and along with ammonia, while succinate was elevated. And when we looked at the lactate metabolism and turnover from samples that we incubated in vitro, we found that the maximum rate of lactate utilization was in most cases always high, and the production rate was low and the maximum utilization rate was high. But there were exceptions in 3 of the low methane yield sheep where the lactate concentrations were elevated, and that was mainly due to high rates of lactate production and a low utilization rate. And when we put in labeled lactate, most of that lactate was retrieved as labeled acetate, and as the main product, and then propionate and butyrate to a lesser extent. So these results indicate to us that the low methane phenotype sheep is linked to butyrate production via lactic acid, and differences in protein and succinate metabolism might also be evolved in the low methane yield phenotype. So what are the conclusions? So just going quickly through these, the in vitro LAB have been shown to have effects on methane production in a strain-dependent manner. We don't really understand whether the LABs or the metabolites affect methanogens directly, but we've now got tools to look at that. In vivo, there's the lack of robust animal trials, so more research is needed. There are a range of different potential delivery options that we can think about if lactic acid bacteria are developed in products, and this can be an alternative approach that might go along and complement the other initiatives such as vaccines and methane inhibitors. And we think that lactic LAB supplementation and the use of silage inoculants can contribute to on-farm management options to reduce methane greenhouse gas emissions. So I'd like to acknowledge the funding from MethLab, which was a face aerogas program in Europe, and also funded by the New Zealand government as part of that funding as well, and the Livestock Research Group of the Global Research Alliance. And Rumining, also funded by the New Zealand Ministry of Prime Industry and managed by the the Ag Emissions Centre under the New Zealand Island Joint Call. And the various people that have been involved in the program along the way are listed there as well. So thank you very much.
Thanks, Graham. So if I can get you to stop sharing, and next I will introduce Catherine and Tanya. You can probably start to share Catherine's presentation. So yeah, next up, I'm really delighted to introduce Professor Catherine Staunton from Teagasc Moor Park, Cork in Ireland. Catherine is the Principal Research Officer, where her work focused primarily on nutrition, microbiology, and the gut microbiome and the interfaces between those areas. Catherine would never mention it herself, but she is actually one of Ireland's most highly cited researchers and has been for several consecutive years. She's been placed in the top 1% of researchers globally in her field, which is absolutely amazing. And I mean, it's amazing to have someone of Catherine's calibre with us this morning. And thank you, Catherine, and for all our other Irish audience members for staying up late at night to attend this webinar. So, Catherine, over to you. Looking forward to your presentation.
Thanks, Sinead. Can you hear me?
Yes, we can hear and see you.
Okay. Thank you. Yeah, so thank you for the nice introduction, and thank you, Graham, for all the background that you supplied, because really a lot of my presentation is already covered effectively by your very nice overview of the MethLab and Rumining projects. So as Graham said, indeed, we have been working together in this area of lactic acid bacteria and their potential to develop methane mitigation strategies in ruminants for the last number of years. First with the MethLab project, which was funded by Aerogas and involved a number of partners across Europe and New Zealand Ag Research Group, and more recently the Rumining project, which effectively continues to mine the rumen for strains that have the potential to be efficacious in terms of reducing methane emissions from ruminants. And so I'm just showing there the MethLab logo and the Rumining logo, kind of demonstrating the sort of overview of the program involving lactic acid bacteria strains that were well introduced by Graham in the previous talk. So next slide, please, Tanya. So global warming and methane. Well, methane is the second most significant contributor to greenhouse gas emissions in Ireland. And Ireland has a target of a 25% lower greenhouse gas emissions for its agricultural sector by 2030. So the objective of the research that we're conducting is to exploit bacteriocin producing cultures to inhibit methane producing bacteria. So here in Cork, we have a particular interest in cultures that have the potential to produce bacteriocins, which are antimicrobial peptides that inhibit the growth of other closely related bacteria, and in this case, to inhibit the methanogens that are the cause of methane production in the rumen. And while methane fuels global warming, cutting methanogens or inhibiting their potential to produce methane in the rumen has the potential to control methane emissions from the agricultural sector and alleviate some of the stresses associated with that sector in terms of methane production. Next slide, please. So the rumen microbiome is a diverse and complex community of microorganisms present that inhabit, not inhibit there as shown on the slide, that inhabit the rumen, and is characterized by high population density and extensive diversity. So 1 milliliter of rumen fluid contains about 10 billion bacteria and 10 million protozoa and 10,000 fungi, whereas the actual rumen contains about 40 gallons of fluid, so has a vast variety and complexity of microorganisms that contribute significant benefits to the host in terms of digestion of the feed intake. But as a byproduct of that activity, methane production, in particular by the methanogens previously described, is a byproduct which is a significant problem in terms of greenhouse gas emissions from the agricultural sector. Next slide, please. As another aspect of the microorganisms present in the rumen are the lactic acid bacteria, which are a beneficial group of microorganisms that are widely used in human and animal feed applications. The lactic acid bacteria, as previously introduced, are found in food and fermented foods, and in the human gut as well as in the rumen. There are many types of LAB, such as lactobacillus, streptococcus, some bifidobacterium can also potentially be included there. And some key facts about lactic acid bacteria is they produce lactic acid through fermentation, which they help to improve digestion and boost the immune system, and are widely used as probiotics in human nutrition, but also in animal nutrition, and also as food cultures for food production. They have the potential to inhibit harmful pathogens, primarily by the production of bacteriocins, which are small proteinaceous compounds produced by lactic acid bacteria, which kill other bacteria, including pathogens like MRSA, as shown on that slide there. Next slide, please. So we looked at the Hungate 1000 collection, which is a large collection of rumen cultures, and mined the culture collection for its ability to produce bacteriocins. Bacteriocins, as I said, are antimicrobial peptides and proteins widely distributed among bacteria and increasingly recognized as alternatives to antibiotics. The Hungate 1000 collection is a culture collection of prokaryotic microorganisms derived from ruminants. A total of 408 novel bacteriocin gene clusters were identified across 308 genomes. And the frequency of putative bacteriocin production within the ruminal strains was 30%, and this number increased to over 70% when encompassing groups of peptides with limited knowledge of antibacterial activity. Among the bacteriocins found were a wide range of niacin and niacin variants. Next slide, please. So the question is, could lactic acid bacteria kill methane-producing bacteria and thus decrease rumen production of methane by being used as either direct-fed microbials or silage inoculants? Targeting methane-producing bacteria via the production of bacteriocins, lactic acid bacteria have potential to inhibit methane emissions in ruminants. Next slide, please. First, to address this question, we conducted a systematic review to look at the potential of silage inoculants to mitigate methane production from the rumen based on available literature. And based on the keywords that we used in this review, including methane emissions, lactic acid bacteria, and there were some other words. We identified, anyway, 10 studies that described relative reduction of methane production compared with control based on in vitro and in vivo studies. In fact, the majority of the studies were in vitro studies, with only one in vivo study shown here. The majority of the studies demonstrated a relative reduction of methane production as a result of silage inoculants with lactic acid bacteria, primarily Lactobacillus buchneri, and additives was the most effective treatment, resulting in an 83% reduction, with the L. plantarum also being effective at causing minus 48% reduction in vitro experiments. One in vivo study shown here involving a Lactobacillus plantarum strain actually didn't result in a methane reduction, but actually a slight increase in methane production. But overall, the results were positive, potentially pretty positive for demonstrating an effect in terms of methane emissions based on the studies already available. Next slide, please. So in our collaborative research with Ag Research, as already outlined by Graham, we developed, based on the work that Graham already spoke about, a league table of bacteria that are capable of killing methane producers in the rumen. And much like the Premier League, we have listed them in order of success there. A lot of them are certainly prominent among the bacteria capable of killing methane producers are those that have the ability to produce niacin. Now, niacin and its variants were very effective in terms of anti-methanogenic activity in vitro. Next slide, please. So, for example, shown here is one piece of information demonstrating bacteriocin producing cultures and the bacteriocin niacin itself were effective in terms of being capable of inhibiting methane production from Methanobrevibacter. And so, as I said, and as Graham said, a large bank of strains were screened and developed for their ability to inhibit Methanobrevibacter. Next slide, please. So we applied two strains, both bacteriocin producers, Lactobacillus plantarum and a Lactococcus lactis, subspecies lactis, capable of producing plantaricin and niacin, respectively. We applied them, these two cultures together, as a starting inoculation to develop a silage inoculated with bacteriocin producing strains. Important to note that both of these strains were not antagonistic towards each other and did not kill each other. Therefore, they were compatible to be used in combination. And when silage was made using this co-inoculation of the two strains, there was effectively no major impact on the quality of the silage produced. So there were similar pH values, ammonia, dry matter, and the LAB numbers in the treatment group were about 1 by 10 to the 9 CFU per gram of silage. Next slide, please. We subsequently went ahead and produced 50-ton quantities of this silage made with and without bacteriocin producing inoculants and fed them to cows who consumed 14 to 15 kilograms per day of the silage, contributing 10 to the 11 to 10 to the 12 CFU per day of the bacteriocin producing cultures. The silage was made in August of 2020, and the MethLab silage is shown there in the purple base, and the control silage was in black. Next slide, please. So we conducted a trial in dairy cows to compare the effects of feeding the grass silage inoculated with the lactic acid bacteria from producing cultures, versus uninoculated grass silage on animal productivity and methane emissions. So 30 cattle were selected and fed for 7 weeks in a feeding trial, where group 1 was the control and group 2 received the inoculated silage. Animal production parameters, including weight, milk yield, were tested, methane emissions, and animal health and rumen microbiome was analysed. The animals were actually maintained indoors for this trial. And as I said, the co-inoculant Lactococcus lactis and Lactobacillus plantarum were inoculated into the treated silage. Next slide, please. Overall, there was no significant reduction in methane as a result of when analysed over the entire trial. But if we looked specifically from week 4 on of treatment, we did find a significant reduction in methane production from the treated animals, where there was about a 5.8% reduction in methane emissions from the LAB treated animal group. Next slide, please. So the outcome of the trial was that LAB bacteriocin strains inhibited methane production. The silage feed could be manufactured containing high levels of the live strains because the strains actually grew very well in the silage during the ensiling process and during the storage and maintained significantly high viability throughout the study. The cows were fed indoors with the strains and methane emissions dropped by about 6% from weeks 4 to week 7, while there was no impact on weight gain, milk yield, and other production traits. In terms of the Irish national herd, which consists of about 6.9 million animals, this 5.8% reduction in methane emissions would be equivalent to the output of about 405,000 animals. Next slide, please. So the continuation of this work in the remining study, where we're mining the rumen for methane reduction using various different approaches, some of which have already been outlined by Graham. We're looking at the role of microbiome in methane production. We're looking at identifying the characteristics of low methane microbiomes. We're looking at testing and enlarging the league table of strains in cows to reduce methane, and also looking at viruses and their potential role in methane production or inhibition of methane production. Next slide, please. So the experimental approach involves a number of different approaches. Like we're looking at a number of strains and have identified a number of strains that are outperforming those previously used in the MetLab study. These are being investigated in vitro in batch culture and Rusitech experiments with our colleague in Chagas Grange, Paul Smith, and his group. We're also looking at alternative feeding strategies. We talked about the silage inoculation, but also investigating direct fed microbials with longer trial durations, looking at strain viability, and also using shotgun metagenomic sequencing. Next slide, please. Just demonstrating the in vitro approaches using the Rusitech, where we're inoculating Rusitech in vitro fermentation with and following fermentation over 21 days. Next slide, please. There are just a couple of pictures, really. Okay, and are evaluating methane emissions, gas production, and volatile fatty acids. Next slide, please. So just some results to demonstrate that inoculation of a particular Lactobacillus plantarum and a different Lactococcus lactis than the one previously mentioned demonstrates that these treatments result in reductions results in increased VFA production, which indirectly reduces methane production. So the L. plantarum showed increased VFA production compared to controls, and also the Lactococcus lactis strain. Next slide, please. So modulating the rumen microbiome to reduce methane, certainly probiotics and direct-fed microbials may redirect rumen biochemical pathways. Hydrogen sinks lead to propionate production, acetogenesis, and nitrate reduction, leading to improved feed efficiency and having less hydrogen for methanogenesis. Next slide, please. And we're conducting further rumen studies in animal trials, which are planned for autumn 2026 to investigate the Lactobacillus plantarum strain in particular in a crossover study design to investigate its potential for longer term methane emission reduction. Next slide, please. So in summary, bacteriocin-producing lactic acid bacteria may transiently influence rumen microbial communities and reduce methane output. There is a need to optimize delivery strategies and maximize their potential to support sustainable dairy production. And by combining lactic acid bacteria with other strategies such as dietary interventions, slow-release additives, or microbiome-targeted approaches, there is the opportunity to give scalable, sustainable solutions for reducing methane emissions. Next slide, please. And just finally, thanks to all my colleagues here in Ireland, especially Michael O'Donovan, Michael Egan, Paul Smith, Pat Dillon, and our colleagues in UCC, Paul Ross and Colin Hill. And big thanks also to the great collaboration we have with Ag Research and Graham and his colleagues in New Zealand. Thank you very much.
Thanks, Catherine. So Tanya, if I can get you to spotlight the team, and we'll get into some of the questions. I see, Graham, you've been answering them in the Q&A, so thank you for that. So please have a look, people, at the questions Graham's been answering. But maybe we'll get into some of the questions that are still to be answered. So Graham, I think this relates to your one, I think, where someone was saying, is there any reason, and maybe Catherine, you may have comments on this, why LAB might have worked better in lambs compared to cows?
Yeah, so I think there's 3 studies which I highlighted which showed effects in sheep, and in one of them, the work by Mala Janathan, she showed that the strains that had an effect in sheep weren't effective in cattle. I don't know why that would be, apart from that a sheep is not a cow. So there's from the work that we've done, we've seen, obviously quite large differences between the LAB strains and their effectiveness in vitro and in vivo. So there's possibly strain differences, and I imagine that those strain differences then behave differently in different backgrounds. So we know there are, the microbiome of a sheep is not exactly the same as a cow. They generally have many of the same functional groups, but there are specific differences which exist in sheep and cattle. One difference is the prevalence of organisms called Quinella in sheep. They seem to be sheep specific, so they're not found in cattle, and that happens to be one of the organisms which is more abundant in one of the rumotypes, which is linked to low methane yield. So I don't have an answer of why they're more effective, but we can guess that it might be related to the composition and the makeup of the microbiomes.
Well, thanks, Graham. Catherine, there's one that Graham's already had a go at answering, but I think, you've obviously got a lot of experience working with monogastrics. And, when we talk about LAB probiotics in the human world, we're really talking about that GI2, but in ruminants, we tend to talk about the rumen. But what about the impact of the hindgut? Is it a possibility that the LAB activities, could be impacting there? And have we really looked at that? Has all the focus been on the rumen, and we've missed an obvious gap in our knowledge here?
Yeah. Well, Absolutely. I mean, there are a number of benefits to LAB in terms of their potential in the gut, and they inhibit, like via bacteriocin production, inhibit certain other bacteria, and of course, as Graham mentioned, the bacteria present in different species are different, and therefore their effectiveness would potentially be different in terms of cattle versus sheep, for example.
Cool. Thanks, Catherine. We have a question about variability. If you look across, a lot of the research around LABs, there seems to be variability, and sometimes they work, sometimes they don't work, sometimes they work a little, sometimes they don't work a little. Any thoughts to what might be causing this variability with these types of products, not just in methane mitigation, but also obviously in animal health?
Well, I mean, for probiotics, obviously, it's important that the strain is alive and that the dose is given at the right effective dose to get the effect. And depending on the strain and the quality parameters used and storage conditions and age of the product, you will find some variation in products that contain live cultures in terms of whether or not they're all fully alive still, or there's some loss of viability. And so the dose is very important. And if the viability is gone down somewhat in the product, then maybe that would account for some of the variation or lack of efficacy in some cases. That's just one potential
Cool.
reason why you might see variation between different
Cool.
uh,
Well,
products.
thanks, Catherine. A question by David. Obviously, some of the results both you and Graham have shown, quite impressive in vitro results. But the question David is asking is, what are the factors that need to be understood to sort of translate those into, I think, in vivo reductions in animals? Any thoughts on that?
Yeah, well, there's as I pointed out, there's a whole raft of in vitro studies, and as David points out, some of them have quite high levels of inhibition, quite impressive levels. So the in vitro is not a rumen. So there's it's a rough approximation of what happens in the rumen. So you're taking rumen contents and you're incubating in the glass container for a fairly limited period of time, and you're asking the microbes in there to survive and behave as normal. And of course, they don't act perfectly as normal because they're not in the rumen, and you're not getting the interaction with the host, and you're not having the continuous removal of end products, which allows organisms to grow optimally. So there's those sorts of things. What we tend to do with rumen in vitros is we take them as a bit of a guide. So obviously, if you don't see any specific effects in pure culture assays against methanogens, you're unlikely to see much of an effect in vitro. And then the next step is to go to in vitro. And then again, if you don't see much of an effect there, then you're unlikely to see much in vivo. So we use it a bit like a funnel um, to cut down, reduce the number of things that we take through to testing in animals. Now, I think what David is asking is how can we make in vitros a better predictor of what happens in animals? And I agree, there needs to be quite a bit of work invested in making rumen in vitros more rumen-like and more predictive of what happens in the animal, because we can't always afford to do large animal trials, and they have to be large. The animal numbers have to be large to be able to detect the sometimes small amounts of reductions in methane that these types of compounds result in. So yeah, there is a need, I think, to improve the quality and the predictability, so to speak, of those rumen in vitros.
Cool. Thanks, Graham. We've got a question from Carl, and not sure if Catherine or Graham wants to take a stab at this one, but do you expect any interaction between LAB treatment and feed additives? Has anybody done that work that you're aware of?
Have they
Um,
do? Yeah.
So I think
Okay.
there's some evidence of co-inoculation with an LAB with a acetate an acetogen, so an organism which can take over the role of methanogens in the rumen by using hydrogen and CO2 to form acetate rather than allowing it to be converted to methane. There's been a little bit of work on those sort of co-inoculation studies, and the one that's been reported showed some benefit of that. I would expect feed additives which improve the availability of easily fermentable carbohydrates in the rumen or in the lower gastrointestinal tract would be able to support LAB activity and persistence in the gastrointestinal tract. So any feed additive which achieved that, I think, would be beneficial.
Cool. Thanks, Graham. There's another two questions, and two of them are sort of similar, and it's really around, are there any consequences of feeding the LOB in grazing animals? And is there, I guess this question is maybe looking at, are there any unintended consequences you see, or inversely, are there any benefits you'd actually see to feeding these types of bugs? Because of course we as humans take them for all variety of reasons. And then there's a question, great, or sorry, question that relates to if you're interested in doing more work. There's someone on the call that has a really good LAB, so I'll direct them to your emails to get in contact if you want to connect with those. But yeah, that first question really, any unintended consequences or actually any positive benefits that you can see with this type of mitigation approach.
Graham, do you want to take it?
Well, the known consequences of inhibiting methanogens is the accumulation of hydrogen. So methanogens exist naturally in the rumen, right? And their role in the rumen is to mop up the waste products of other organisms. And one of the really important ones to control is hydrogen. Now, if hydrogen accumulates in the rumen and its concentration gets too high, it can have negative effects on fiber degradation via the action of fiber degrading microbes, which I think is the point of that person's question. So the thinking around that is to use LABs to inhibit methanogens, but also to provide a situation where we encourage the growth of alternative hydrogen users. I've mentioned acetogens being a potential user of hydrogen, but there are also a number of other organisms in the room which can use hydrogen in one way or another. Some of them use it to reduce sulfate, and that can redirect hydrogen towards that process. Nitrate reduction is also another endpoint for hydrogen and the electrons that hydrogen carries. And the other potential use of hydrogen is via reduction of fumarate through to succinate. Now that's probably the most beneficial one, because if you reduce fumarate to succinate, then succinate will then go through to propionate, which is of benefit to the nutrition of the animal. So yeah, we realise there are potential consequences of interfering with the methanogen use of hydrogen, but we think that we can either add other organisms to alleviate that, or once the hydrogen concentration gets to sufficient levels, these other organisms will kick in and help dispose of that hydrogen via other routes.
Okay, we've got probably only about 2 minutes left, and maybe a quick rapid round to see if we can finish off the questions that have been asked. Has there been any focus on early life intervention with LAB for methane modulation or plans to? I know there's quite a lot of work going on internationally, but in your groups, are you focusing on early life?
Certainly in the research planned at Grange, Chag's Grange, led by Paul Smith, there are plans to look at early life interventions. So yes.
Cool. Thanks,
And we've
Catherine.
done some commercial work with early life intervention with LABs too.
Cool. Thanks, Graham. I think this could be a very quick answer. Why not just feed niacin instead of the LIB?
That's been tried, but niacin is susceptible to the proteases in the rumen, so its half-life in the rumen, I think, is compromised, and so it doesn't appear to be useful being used by itself.
Cool. Quick question for Catherine. With the silage inoculant study at Chagas, do you think it was the LAB itself having the effect, or did it change the silage itself in terms of nutritional quality?
We tested the nutritional quality, which was similar between the control and the inoculated silage. So we do believe that it was the impact of the live LAB cultures that had a direct effect in vivo, yes.
Cool. And someone's asking a question on biochar. That's not for today, but yes, there are people looking at work in terms of biochar, in terms of a methane mitigant. Most of the research results related to feeding biochar in enteric fermentation have been not positive, but there are certainly people globally working in that area. Okay, that brings us to a close. That was a fantastic webinar. A big thank you to Catherine and to Graham for their presentations today, and also their answering of the questions. Also, thank you to our audience, particularly those that have stayed up late tonight to watch this webinar. And with that, just wish everybody a very good day or evening. Yeah, and please tune into the next one. Thanks all, and slán a bhaile, as they'd say.